Um, yeah. Hi, everyone. I'm Sehi. I'm a postdoctoral research fellow in Harvard Medical School. So what is visualization authoring? Visualization authoring is a task to transform data into a visual representation, which is an essential but non-trivial process for making sense of data for any data-driven fields. Researchers have extensively studied various techniques, interfaces, and tools to understand how we can best support people authoring visualization in an effective and efficient manner. Here are several examples that we identified through paper review, which all have trade-offs in their usefulness. For example, template and shelves are easy to use, but they are limited for making highly custom visualizations. Natural language interfaces and visualization by example are user-friendly but less accurate compared to manual approaches. Wiring code is likely most expressive, but it comes with a steep learning curve. In the genomics field, only a few of these techniques has been adopted, and the usefulness of many other techniques remain unclear. So this leads to our research question. What visualization authoring technique can best support the diverse group of genomics experts and in which tasks? To address this question, we conducted 2 studies with genomic experts. We first wanted to understand how diverse genomics experts actually author data visualization and what are the difficulties. We conducted a semi-structured user interview to identify their visualization authoring workflows, tasks, tools, and challenges. After conducting the user interviews, we performed qualitative analysis with 2 of the paper authors. Through the analysis, we identified 5 distinct user groups of genomics visualization authors based on the focus of their studies, the automation level of their analysis, and target audience of their visualizations. We used these 5 personas to characterize different characteristics of user groups and also used it in the following study. So in the second study, we conducted an exploratory study to understand how diverse genomics experts ideally want to author visualizations. We addressed this question by collecting preferences and perceived usefulness of 6 authoring techniques using a study material called visual design props. Design prop is similar to low fidelity prototypes that contain visual elements and interactive features, which is helpful for making people to reflect on and share their experiences, feelings, and attitudes. Using props in our study enabled users to better engage with 6 authoring techniques. We created 2 sets of visual design props. One is for the training session to help participants experience and learn each of the 6 authoring techniques. And the other was for the main session to guide them through realistic scenarios to help them provide insightful feedback during the main session. So here is a result. In this summary chart, each column represents a specific task given to users. The color of the bar represents the 6 different authoring techniques, and the height of the bar represents the number of participants who preferred the corresponding techniques for given tasks. Interestingly, none of the participants selected a single technique. As you can see, there are many bars with different colors. And participants noted that different authoring techniques were better suited for different tasks. We further look into the result by different user groups. Again, 8 columns represent the preferences for authoring techniques by tasks, and the bars on the right highlight aggregated preferences across all tasks. For example, you can see the yellow bar is taller than the others, indicating that code editor are the frequently mentioned method as a viable option for all tasks. We can observe several interesting patterns across user groups and tasks. For example, biologists were generally reluctant to use techniques that are less familiar in genomics, such as shelf configuration interfaces. Also, all user groups were willing to use unfamiliar techniques when they are asked to perform difficult tasks, such as authoring coordinated multiple views. This shows the potential for adopting novel visualization authoring techniques to support more complex but important authoring tasks. Through interview, we further identified the perceived usefulness of 6 authoring techniques, and here is an example about natural language interface. Participants frequently noted that it is often difficult and laborious to describe what they want to achieve, as one participant mentioned that it is very, very cumbersome. One of such examples was describing a custom visualization that does not have a clear name. So people have to describe individual visual mapping to indicate their intention in their authoring tasks. I think we need to be careful about adopting that large language model without clear understanding of target visualization tasks based on our feedback from the people. In our paper, based on the challenges and benefits of various authoring tools, and through iterative discussions with the authors, we developed 5 design implications for genomics tools. Since our user studies show that multiple techniques are preferred for different tasks by all user groups, we think tool designers should consider combining multiple authoring techniques to meet the diverse needs. I think genome browsers that support computational notebooks to enable easier data processing are good examples of combining template tool and also code editors. And I think there are many more opportunities in combining other techniques as well. Given the time limit, I won't go into all details, but for more insights into building genomics data visualization authoring tools, please read our paper. So in summary, through 2 user studies, we identified 5 personas that describe distinct group of genomics data visualization authors. We identified the benefits and challenges of using 6 authoring techniques for performing different tasks. And lastly, we identified 5 design implications and research opportunities for visualization authoring tools. With that, I would like to thank you for your attention, and I'm happy to take any questions.
Somebody's got to ask a question. So for your biologist persona, biologist is a pretty broad term.
Yeah.
How did you define biologists? Are these traditional bench biologists and spent most of their time in the lab? Because I was a little surprised by the height of the code bar for biologists.
That's true, yeah. So I think I need to clarify that. So our study only looked into 20 participants, and many of them had the computational skills. So maybe the name of the persona might not be representative, but we kind of named the persona based on the focus of their work, whether they are more leaning toward asking questions about the biology, or they are more interested in computational problems, how much they use automation and audience of the.
It'd be interesting to get as another persona a bench biologist who are generally not as computationally aware. So it'd be just an interesting that you'd see a little balance where you wouldn't have so much of that code. So thank you.
I think that is a good point. Yeah, I think we can we hope that in the future people can expand the persona that we have. So I think that is a good point.
Let's thank our speakers.